Till recent times, late blight of potatoes was reasonably simple to manage. But in the late 1980s, new populations of Phytophthora appeared, with the combined genetics from strains from both North America and Europe. Now the new strains of Phytophthora are more aggressive. Epidemics are more severe, with earlier infection and shorter life cycles. Reason enough to think about a reliable, effective and long-lasting protection for potatoes. Assurance for the grower, for potato quality and yield. Nature gave us the inspiration for a perfect protective shield. We have adapted this knowledge for potatoes with the new active ingredient fluopicolide, an innovation of research and development by Bayer Crop Science. Part of the new chemical class acylpicolides, fluopicolide establishes a novel mode of action in disease management. This agent is highly effective against the pathogen at all key stages in its life cycle. The consequence? No chance for late blight. Its activity against motile zoospores is dramatic. Zoospores stop moving immediately on contact. Within a few minutes, they swell and burst. Sexual reproduction has allowed Phytophthora to combine the attributes of various genotypes. The most fit and aggressive evolve, survive and prosper. The result is that more pathogenic, faster spreading and more prolific strains are found around the world. Phytophthora is well adapted to all environmental conditions where potatoes are grown. Under warm conditions, direct germination allows the sporangia to germinate directly. Under cooler conditions, indirect germination causes the sporangia to form and release multiple zoospores. Both routes are effective in infecting the plant. In both cases, fluopicolide shows the same sweeping success. In mycelia, or zoospores of Phytophthora, spectrin-like proteins form a net along the cell membrane. This net is anchored on the membrane and strengthens it. At the same time, this net is the point of contact for the skeleton of the cell, called the cytoskeleton. When fluopicolide enters the cell, it causes disorganization of the spectrin-like proteins. The net is torn to pieces. The membrane is weakened and the cytoskeleton becomes unstable. It detaches from the membrane. For our global player, Phytophthora, this is a catastrophe. Mycelia stop growing and zoospores swell and burst. That means no chance for late blight. Most of the fluopicolide stays on the plant surfaces and forms a shell of protection. It provides a reservoir of product that moves strong translaminar properties. When applied to the base of the leaf, it also moves into the leaf tissue. When applied to the upper side, it also moves to the lower. That means complete protection for the plant, for leaf